Hello friends, today we are going to discuss the new topic from the spectroscopy paper, and the title of this topic is nuclear magnetic resonance. In short, it is called as NMR spectroscopy. Earlier we have discussed the first important spectroscopic technique, which is called as infrared, that is IR spectroscopy. So we have already discussed the scope or the importance of IR spectroscopy in organic compound. IR spectroscopy is used to detect the functional groups that are present in the organic compound. Now today we are going to discuss the another spectroscopic topic, that is nuclear magnetic resonance spectroscopy, and the important scope of this topic is to assign the structure for the unknown organic compound. Now here this NMR spectroscopy, as far as NMR spectroscopy is considered, these are the three guys who have contributed hugely in the NMR spectroscopy. So this fellow is Rabi, this is Purcell, and this one is Bloch. So these three guys, out of these three guys, Purcell and this Bloch received Nobel Prize in 1952 for their greater contribution in nuclear magnetic resonance spectroscopy. And here you can see the image of the NMR spectrometer, which has the capacity 1 gigahertz, which is there in France. So this is high capacity NMR spectrometer, which is present in the France. Now let's discuss the scope of this NMR spectroscopy. Scope in the sense, what is the utility or what is the importance of this NMR spectroscopy in organic chemistry or in chemistry, we can say in general. So the first scope, as this topic is involved in the BSc 3 syllabus, so it is a part of curriculum. So we have to study this NMR spectroscopy, and after studying this topic we can determine the structure of organic compound. So the important application of the NMR spectroscopy is to know the structure of organic compound. If we record the NMR spectrum of unknown organic compound, by analyzing that NMR spectrum, we can be able to assign the structure for the organic compound. Now organic compounds can be isolated from different natural sources, that may be from plants or animals, or it may be prepared artificially in the laboratory, and after isolation or after preparing the organic compound, the very next step is to know the structure of that organic compound, because once we know the structure of organic compound, we can come to know what sort of reactions that compound can show, which functional group is present in that organic compound, what is its chemical activity, biological activity. All these aspects comes sorry, when we know the structure of organic compound. So structure determination, which is the important application of the NMR spectroscopy, and that is the first and important scope of this topic. So the students who are not taking the higher education after BSc, if some students are interested to have some short term course and they are willing to join for the job, then they can take the short term course of NMR spectrometer, how to operate the NMR spectrometer. So after completion of this course, they can get the job of NMR operator, which is a good salary job, and there you have to analyze the organic compounds for the structure determination. So this is about the scope of this topic. It is used to study the structure of organic compound, and if the students are willing to do the jobs, then they can get the job of NMR operator. For that, obviously they have to complete they have to take the training of how to operate NMR spectrometer. The cost of this NMR spectrometer is in crores, therefore great care should be taken while operating this instrument. Now let's come to the next slide, and the topic NMR spectroscopy consists of all these points. So the first, we are going to discuss the introduction of this topic. Secondly, we are going to discuss the principle of this topic. Then instrumental part, that is instrumentation of this NMR spectroscopy. Then magnetic and non-magnetic nuclei. So this is very important point, and most of the times in the previous exam the question has been asked, write a short note on magnetic and non-magnetic nuclei. So that part we are going to discuss in the due course of this topic. So magnetic and non-magnetic nuclei, for the time being, just I want to mention here, magnetic nuclei are only NMR active, non-magnetic nuclei are NMR inactive. Remember this point. The next point is chemical shift. So this point we are going to discuss with respect to its definition, measurement, calculation, factors affecting on chemical shifts. So this is the chemical shift, which is the scale that is to be used while plotting the NMR spectrum. So chemical shift can be expressed in terms of delta as well as in terms of tau scale, that we will see in the further session. The next point is shielding and de-shielding. So shielding and de-shielding. So again, this is the important point as far as the examination is considered. Most of the times the question has already been asked in the previous exams on this point. Write a short note on shielding and de-shielding effect. So again, we are going to discuss this point in detail in the coming sessions. The next, peak integration. We are getting different peaks in the NMR spectrum. When we record the NMR spectrum of the organic compound, we can have a number of peaks in that NMR spectrum. So we have to integrate these peaks, and the peak integration is essential to know the peak area of the protons. Now what is the use of this peak area? What sort of information we can obtain by knowing peak area after peak integration? So peak area gives the information about the number of each types of protons. So again, we are going to discuss this point in detail. The next is merits of TMS as PMR reference compound. So merits or the advantages of TMS. What is the long form of TMS? Tetramethylsilane as PMR reference compound. So what is the long form of PMR? Already we have discussed. NMR means what? NMR, nuclear magnetic resonance. Here PMR stands for proton, and proton is the nucleus of hydrogen, and when we consider the proton as a nucleus or the nucleus of hydrogen, which is also called as proton, then that technique, that spectroscopic technique is called as PMR spectroscopy. So the next point, coupling constant, which is important to know the types of protons, and it is expressed, it is denoted by capital J letter and expressed in hertz. The next point is types of coupling constants. Coupling constants are of different types, and we are going to discuss the different types of coupling constants in the coming sessions. Then the next point is the spin splitting, and the last point is applications of the NMR spectroscopy. So here these are the points that we are going to discuss under this topic. So instead of NMR, nuclear magnetic resonance, we are going to use the words like proton PMR, that is proton magnetic resonance spectroscopy. So here we are going to consider the nucleus of hydrogen, which is nothing but the proton, and hence it is called as proton magnetic resonance spectroscopy. As you know, organic compound is the compound which consists of carbon atom. So along with carbon atom, hydrogens are also there. So from the hydrogens, from the number of hydrogens which are attached to the carbon atoms, we can find out the functional form of these hydrogens, and this can be done in NMR spectroscopy. Once we know the functional form of hydrogens, we can assign the discrete units or discrete parts of the functional forms of carbon and hydrogen, and we can assemble these units so as to get the structure of the organic compound. So this is the syllabus that we are going to discuss in NMR spectroscopy. Now let us see the meaning of nuclear magnetic resonance spectroscopy. The first word is nuclear. So this word is related with nucleus, means there must be nucleus of some atoms involved in this technique. Now, as I mentioned, magnetic nuclei are only NMR active. So what is meant by magnetic nuclei and non-magnetic nuclei? We will discuss that part in the next video. The next, after the first word nuclear, the second word is magnetic. So magnetic means there must be some magnetic field, magnets and magnetism. So there must be relationship, or there must be some magnetic field involved in this technique. The third word is resonance. So resonance is nothing but the matching of frequencies. So what is the meaning of this matching of frequencies? Suppose particular FM radio station is sending radio frequencies of certain wavelength, and if we tune in our mobile or radio set the same frequency, then the transmitted frequencies of the FM radio station will be absorbed, or they will get matched with the frequencies that are adjusted, that are tuned in our mobile phone or in our radio set. So whenever there is matching of the transmitted frequencies and the frequencies that are tuned in the receiver, this is called as resonance. So this resonance phenomena is very important in nature also. So that is the another part to discuss this resonance phenomena in insects and other animals also. So that is not the part of this topic, but here resonance means matching of frequencies. In life also, unless we have unless the two persons have the matching frequencies, they can't come together. So that is the natural thing. The last word is spectroscopy. Already we have understood the meaning of spectroscopy in the first topic, IR spectroscopy. Spectroscopy, it is the interaction of matter with radiations. When matter interacts with radiations, then that technique is called as a spectroscopic technique. So in NMR spectroscopy, which types of radiations are used? Radio radiations are used, having frequency 100 to 600 megahertz. So this is the radio frequency that are used in NMR spectroscopy. So which samples are used in NMR spectroscopy, or which samples are analyzed by using this NMR spectroscopy? So here solids, liquids as well as gases can be analyzed for their structure determination by using this NMR spectroscopic technique. It is very versatile spectroscopic technique. It is a unique spectroscopic technique. After recording the NMR spectrum of the sample or the organic compound, we can recover that organic compound for further analysis. Further, we can carry out the kinetic as well as structural studies by using this NMR spectroscopy. Again, we are going to discuss the applications of NMR spectroscopy in the end of this topic. Now let's see what is NMR plot. NMR plot means what is NMR spectrum. So you might have seen the cardiogram. In cardiogram, number of peaks are there. Those same pattern in similar way. It is not exactly similar, but similar types of peaks are observed in NMR spectrum. So what is this plot? It is the plot of peak intensity on y-axis versus chemical shift on x-axis. So chemical shift is expressed in two units, delta scale and tau scale. So delta scale, it starts from right to left, 0, 1, 2, 3, 11, 12, etc. So this is expressed in terms of ppm. So 0 delta, 1 delta, 2 delta, 3 delta. So this is delta scale. So when we go from left to right, so here 0, 1, 2, 3 up to 11. So that scale will be called as tau scale. So right to left, that is delta scale, and left to right, that is tau scale. So we are using delta scale. Right to left, it is increasing from 0 to 11 or 12. Now what is this upfield or shielded, and what is the meaning of this downfield and deshielded? The protons. So remember one thing, hydrogens are present in every organic compound. The nucleus of hydrogen, if you consider the hydrogen atom consists of nucleus, and in that nucleus one proton is there. Around that nucleus one electron is revolving. So for the time being, if we keep aside the electron, if we remove the electron from the hydrogen atom, then there will be only nucleus, and that nucleus consists of one proton only. So in another words, we can say that the nucleus hydrogen will be considered as proton, or it can be called as proton. So organic compound consists of different types of hydrogens. So these hydrogens henceforth we are calling as protons. The protons which are showing the peaks in this region, that is in the region of low delta value. So these protons will be called as shielded protons, and the protons which will show the peaks at higher delta value will be called as de-shielded protons. So shielded protons, they show the peaks at high field side. Low delta value means high field side, and higher delta value, this higher delta value means low field side or down field side. So that is the difference. You should note it carefully. So low delta value means high field side, the protons will be shielded protons. The higher delta value means low field side, the protons will be de-shielded. Now what is this TMS and CdCl3? So these are the reference compounds or the standard compounds that may be used while recording the NMR spectrum of organic compound. TMS, as I told you, its long form is tetramethylsilane. So this tetramethylsilane consists of 12 equivalent protons, and it is used as a reference compound in NMR spectroscopy. Why it is used as reference compound? It has many reasons, so that part we will discuss in the due course. And a question was frequently asked in the exams: why TMS is used as a reference compound in NMR spectroscopy? CDCl3 can also be used as a reference compound. CDCl3 means deuteriated, D means deuterium, deuteriated chloroform. Actually it is CHCl3, H is replaced by deuterium, therefore it becomes a CDCl3, and hence it is called as a deuteriated chloroform. So either of these two compounds can be used as reference compounds while recording the NMR spectrum of organic compound, but most of the times TMS is used as a reference compound. And TMS, as I told you, it consists of 12 equivalent protons, and the TMS shows a PMR peak due to 12 equivalent protons, and the position at which TMS shows a peak singlet, that is called as singlet, single peak, that position is considered, or that scale is considered as 0 scale. So there starts, so this is the reference from which we can record the chemical shift. So suppose a particular compound shows a peak at 1 delta value. So that means it is from 1 delta scale from this TMS. The position at which TMS shows a peak is considered as 0, and from this scale, the delta scale will go on increasing towards left side. So this is the NMR plot. It is nothing but the chemical shift in terms of delta scale on x-axis versus peak intensity on y-axis. Now let's take the example of ethanol. How the NMR spectrum of organic compound looks like. So this is the structure of ethanol. This is the ethanol NMR spectrum of ethanol under low resolution, and this is the NMR spectrum of same compound, that is ethanol, but under high resolution. Now let's see what difference is there between these two NMR spectra. So the first important thing that you should remember is that hydrogens from organic compound cannot be differentiated from one another by chemical test. All hydrogens that are present in organic compound cannot be distinguished by chemical test, but here by using NMR spectroscopy we can distinguish the types of hydrogens that are present in organic compound. Now let's consider this ethanol. Ethanol CH3, CH2 and OH. Structure of ethanol CH3, CH2 and OH. Now these three protons, instead of hydrogen we are using the word proton. Hydrogen means proton. We are considering only the nucleus of hydrogen, and it is called as proton. So these three protons which are shown by red color are of one type. These blue colored two protons are of second type, and this green colored single proton is of third type. So in case of ethanol contains three types of protons. So three types of protons are present in ethanol. This can be proved only by PMR spectroscopy. So as we are considering protons, we are using PMR spectroscopy wordings. Okay, now let's take the PMR spectrum of this ethanol under low resolution. So these are the two spectra of ethanol under low resolution and under high resolution. If you compare these two spectra, you will find there is a difference. So under low resolution we are getting 3 peaks. As 3 types of protons are present in ethanol, we are getting 3 peaks for 3 types of protons. here also we are getting 3 peaks. This is first, second, and this one is third. But this peak is a single. Here it is also single. This peak is single under low resolution, but this peak is splitted into 4 peaks, which is called as quartet under high resolution. This peak is due to CH3 protons, 3 protons, and it is singlet. Its peak intensity is more because it is obtained due to 3 protons. Its peak intensity is lower because it is obtained due to 2 protons. Its peak intensity is lower as compared to this because it is obtained due to 1 proton. So here these 3 protons are showing a single peak under low resolution, but under high resolution these 3 protons are exhibiting a triplet. Triplet means 3 peaks are there. Here you can see 3 peaks for these 3 protons under higher resolution when we record the NMR, PMR spectrum of ethanol under high resolution. So this is the difference between low resolution and high resolution. So why this difference is observed when we go from low resolution to high resolution, that we will discuss in the next sessions. So this is about the structure PMR, general PMR spectrum of ethanol. So you can get the idea of the PMR, how PMR spectrum looks like. So in the due course we will discuss the PMR spectrum spectra of different organic compounds, and there is question not frequently but occasionally such type of question is asked. Draw the PMR spectrum of acetaldehyde. Draw the PMR spectrum of acetone. So you have to attempt this question, you should know all the theoretical part of the PMR spectroscopy. Unless you know the theoretical part of the PMR spectroscopy, it is very difficult to draw the perfect PMR spectrum for particular compound. So for the time being, this is the first video that I am sharing with you, and if you have any doubts or queries, you can contact me. Thank you very much.